it's going to be kicked off by Shigehiko Hayashi, and he's going to be talking about chemical reactions. Sorry, yes, it's backwards. So it was going to be QMMM simulations and dynamics of robots. Yeah, he's mine. We're going to talk about this.
But I'm going to talk about QMMM simulation.
Okay, I'll stay up 5 minutes to go.
Okay. First of all, I'd like to thank the organizers for giving me this opportunity to visit to Australia and to present our study. So I'm going to talk on the hybrid QMMM type simulation on fractional protein production. So we are researching chemical reactions in protein. So why we think that to investigate chemical reactions in protein is important is because the chemical reactions play an important role in biological functions of energy conversion and regulation. These are the examples. The left one is photoreceptor. This is a light-driven. This functions as light-driven photon pump. So this protein absorbs light, and by using the energy of this photon, this proton pumps one proton from inside the cell to the outside to create photon gradient across membrane. I don't show the membrane here, but this is membrane protein. And during this pumping process, there are several chemical reactions involved. The first process of this light-driven proton pump is photoisomization of this contour. This is photo reaction, this chemical reaction. And also the proton translocation is done by several sequential proton transfer processes. Of course, proton transfer processes involve bond formation and bond breaking. So it's chemical reaction. The second example is FITPH. This is actually a molecular motor. By using the chemical energy of hydrolysis reaction of ATP, this enzyme rotates its subunit like this way, which can be seen now by single molecule experiments done by Noji at Osaka. So this is a really fascinating system, and one important aspect of this kind of functionality is there are multi-scale features. For example, chemical reaction event takes place in local. It's kind of local event. So, for example, hydrolysis of ATP is just breaking of this bond. But this kind of local event eventually coupled to this kind the huge conformation global motion. This is what we want to know. Then the mechanism of this kind of multi-scale coupling is what we want to know. This is a method. But at this moment, anyway, it is important to understand the chemical reaction mechanisms in protein environment for these functioning processes. So those are the methodology which we could use to investigate protein functions. First one is molecular dynamics simulation. We heard about that a lot. And this is standard method to investigate protein systems. This method can treat large protein systems and also trace dynamics. But this system utilizes empirical force field or molecular mechanics, MM. So this empirical force field doesn't have any explicit description of electric states. So you cannot describe chemical reactions which involve explicit electron structure change. Okay. The second one is molecular orbital theory. We have okay. So this is also we have about that. And this method solves electron function. So we can calculate, for example, transition state of the reaction, which cannot be described by molecular mechanics force field. However, this method costs a lot. requires huge computational cost. So it is not possible to directly apply to entire protein system to calculate chemical reaction. So in order to overcome those difficulties, people, including us, have been using hybrid QMMM type simulations. In these simulations, reactive moieties are described by ab initio QM method. And surrounding protein environment is treated by MM to take into account effect of protein environment on chemical reactivity, which is essential for understanding the enzymatic activity. Okay, so this method saves computational cost a lot. And finally, we can calculate, for example, dynamic process on excited state in protein environment directly. I'll talk about this. This is okay. So those are the reaction protein systems we have studied so far. This is a F1 ATP synthase. I brought a poster there, and I don't talk about this in this talk. But if you are interested in, so please come to my poster. And also we did some quinone reduction in the SPB enzyme, but I don't talk about that. Today I'm going to talk on, I concentrate on the study on photoisomerization dynamics of retinal in rhodopsin, which is, I believe, relevant to the issues in this conference. So let me introduce first what the retinal proteins are. So we are I'm going to talk about proteins, two proteins which are in retinal protein family, which is also called rhodopsins. So rhodopsins are photoreceptor, and these are the membrane proteins. And the proteins consist of 7 transmembrane helices like this way. And most importantly, retinal chromophore is bound in protein as a chromophore to detect light. So this is the chromophore retinal. The chromophore retinal has polyene chain structure, 6 conjugated double bonds, and one end is attached to lysine via shift base. This CN double bond is the shift base, and usually this shift base is protonated and positively charged. Retinal protein function has various functions in various living organisms. For example, this is a rhodopsin in retina of the eye, which is responsible for the vision. And also there is a bacterial rhodopsin called BR, which is a light-using proton pump. So interestingly, those various kind of functions are all triggered by photoisomerization of the chromophore. So you can see the rotation around this one. This is photoisomerization. Okay, this photoisomerization of retinal is known as one of the fastest reactions in nature. For example, if we are photoisomerization complete within 500 seconds. And also, this reaction is very selective. Since the retinal chromophore has polyene chain structure, there are several double bonds which are capable of undergoing photoisomerization. In fact, in methanol solution, there are 3 double bonds. The isomerization takes place at any one of those 3 double bonds. However, in beer, isomerization takes place only around this double bond. So reaction is very selective. this kind of ultra-fast rate and high bond selectivity furnishes those proteins with high sensitivity to incoming light. So it's really important for this functionality. In order to investigate this kind of mechanism, many ultra-fast experiments have been done so far, and recent progress in ultra-fast spectroscopy has attained 10 femtosecond time resolution or shorter time resolution, and made it possible to detect actually the vibrational mode of wave packet in real time. For example, this is a sample of signals measured by this slide group, and here you can see the clear oscillation on sample of signals, which is corresponding to the vibrational motion, wave packet motion. Okay, so this kind of ultra-fast spectroscopy has revealed pretty much important phenomena for the photoreception in rhodopsin. In rhodopsin is the visual receptor. In rhodopsin, photoreception completes within 210 seconds. And they measured the pump signal, and they found that the oscillation is observed in the product state, which means during the photoreception process, the vibrational coherence is kept even under the strong thermal noise of the protein environment. This is quite unusual for this kind of condensed phase system. And VR has also the similar coherent dynamics in the excited state. However, in the formation of the ground state of the photo product, this kind of vibrational coherence is lost. So these are the characteristics of photoreception. So in protein, waves are fast, faster than solution, and in protein, there are strict bond selectivity, whereas there is no bond selectivity in solution. And also quantum yields are high in protein. Dynamics, in terms of dynamics, the rhodopsin has coherent dynamics, whereas VR has incoherent dynamics. So we want to know what is the underlying dynamics which explain these characteristics. So the problem is that in order to simulate this kind of excited state process, we have to one method we could use is MD simulation. However, it's really difficult to construct potential energy surface on excited state because excited state has really complex nature of the potential energy surface. So what we did was we used QMM method to directly calculate the molecular dynamics. So this is a molecular dynamics simulation of excited state by using the QMM Hamiltonian. So here you can see the isomerization, and here and everything we are moving smoothly. And what we found was that this was done in 2003, it's old stuff. And what we found that the process involves really complex molecular motions, like dynamic couplings between vibrational modes. So the isomerization is not only the rotation around double bonds, but it involves large distortion on wagging mode or bending mode. So I'm going to show you the same. So we have rotation here, and also we can see pretty much large group motion, out of plane motion here. So this is this energy profile. So you are here, and also, okay, there is a crossing to the ground state, and it goes to the product state. It takes a long time actually, and actually at that time we don't have any good computational resource for this kind of calculation. So we made the chrome for QM parts short, and but it but it allows us to calculate many trajectories. And these are the ensemble of trajectories. I'm plotting time evolution of energy differences between excited state and ground state. So these are the which are corresponding to emission and absorption energies. So this is the time. Okay, so and essentially these vertical lines correspond to the rotational motion. So we have isomerization like this way. And this is and here is the crossing region where in this region the rotational angle is almost perpendicular. And then after the crossing, some trajectory goes to C13 sheath and some trajectory goes back to for trans. So in this trajectory, we found that photoisomerization is really non-selective. We found that photoisomerization takes place only around this bond, where there are two double bonds which are capable of undergoing isomerization. And also we found that unidirectional isomerization. So also we found that the isomerization is incoherent. So here you can see that each those individual photoisomerization events are distributed widely in time from 110 seconds to 510 seconds. So which is in good agreement with experimental findings. So those characters are actually coming from protein. So in order to see that, we repeated this calculation without protein. So we don't have any protein here. So these are the trajectories. So what we found was that all of the trajectory completes within 112 seconds, which is much faster than trajectories in protein. So it's kind of coherent. And also we found that the hot products are inhomogeneous. We found that there are several trajectories which have isomerization around different levels or different directions. So which means that thermal fluctuation could provide inhomogeneous product formation. However, the protein blocks that kind of wrong pathways and funnels them into the proper one, which properly initiate the functioning process later. Okay. So the good thing of this kind of calculation is that we can calculate naturally the time evolution of emission spectra, which can be which has been actually measured by ultrafast experiment. I don't go into details, but we can identify we can find the good correlation, and we can identify the underlying molecular motions of the molecular motions underlying this ultrafast phenomenon spectrum. So we applied this method to visual receptor adoption. So this adoption has photoisomerization from this bent form to this straight form, and this actually the time constant is 50 seconds, and less than it completes within less than 210 seconds. So this is ongoing, and right now we include whole polyene chain in the QM system, and we use Shigehiko Hayashi. And it takes 4 hours to step forward, one step forward. So it's really very calculation. But we could so far 13 points, but I'm showing that 30, but I'm going to show you 14, the result of 14 trajectories. So it turned out that the photochemical reaction dynamics in rhodopsin is completely different from here. So all of protein trajectories complete isomerization within 110 seconds. So 110 seconds in DR is here, so it's very coherent. And also, and the decay rate is something 50 10 seconds, which is in good argument with the experiment. So why this is so fast is the following. So we plotted the time evolution of dihedral angles. So the red line is the dihedral angle of this isomerizing bond. So this increases smoothly. At the same time, actually, this single bond is moving largely. And we found that this strong coupling between those rotational motion actually accelerate the photoisomerization process. And this is kind of extended flattish motion. And this is a mechanism which that gives the rhodopsin that gives fast reaction and dynamics of rhodopsin. Okay, so I think I skipped this using the schematic view, but I will skip. Actually we can calculate relaxation in photo product, which actually accompanies with blue shift of absorption spectrum. We can calculate absorption spectrum, time-dependent absorption spectrum in this process by using quantum QMM simulation, and we calculated the excitation energy along trajectories and accumulated them, and we found that we really succeeded in producing the time-dependent increase of excitation blue shift. Increase of excitation energy which are observed in experiment. So these are the conclusions that I think this, and thank you for your attention.
Do you have any questions?
Yeah. So actually, I have a quick question. That instead of putting this retinol, that is the chromophore, in this protein, if you just consider the outside dielectric constant, like change the outside dielectric constant, did you get any kind of change in the dynamics?
Oh, I think yes, because this process, actually I didn't explain that, but this process involves really complex curve crossing, could involve really complex curve crossing on the excited state between S1 and S2. And S1, the energy difference between S1 and S2 is really is depending on polarity or electrostatic interaction. So in that sense, if you increase, I think if you increase polarity, you will get slower reaction rate,
right?
Because
that's
it
why
goes
in water it is lower.
That's right.
Pure water.
That's right. That's right. That is one explanation for that.
Are you familiar with Ursula Ruffelsberger's?
Yes.
Yeah. So that's kappa and IDFT, if that's right. Yeah, can you give us the pros and cons of the two approaches?
Actually, they used not time-dependent. I think it's kind of open shell type DFT stuff. And I read her paper, and I think they couldn't get isomerization at 300 Kelvin. And they increased 500 Kelvin. And they increased temperature of chrompa to 500 Kelvin. And I think the potential curve described by the DFT method is not that good. I think this is actually I would say it's obvious. It's really complex. It's really okay. In order to describe curve crossing process, you really need to use flexible wave function. But their method is just simplified, and I don't think it's okay. And also, the curve and energy difference at the curve crossing region is 15 kilocalories or something like that. It's really huge. You never get such kind of fast conical intersection crossing for that kind of 15 kilocalories per mole.
You've just described all my prejudices exactly as I would have said them.
Several questions. Number one, what's really fundamental reason this coherence loss for BR? I mean, time scale not that much. One is 100, one is mesothelioma. To me, it's not that great difference. But how can be one is coherence preserved so well, another is totally lost?
Okay, so
you say something. What's really fundamental there?
I think I did that. Sorry. This is my guess. Okay. So okay, the potential energy profile is something like the downhill path from a mountain, single big mountain, like the famous mountain in Japan. Okay, so for example, there are three paths to the basis from the top, and which are virtually corresponding to those three paths for defined confirmation.
I mean, if you go back to that particular slide, I mean, from an organic chemistry point of view, you could say that you have a cysto-module, so that's isomerized into a trans, so that's a low energy pathway. Here you have a trans, effectively, and so if you isomerize that, then it goes to cis, so that's a higher energy pathway.
Actually, in gas phase and in methanol, it's, we can have those kind of atomizing pathways. Yeah, we can have that kind of pathways, but essentially protein blocks those pathways.
Right.
Yes.
Yeah.
In methanol, in homogeneous environment, we can have smooth pathways around here.
But every time you only have one carbon, right? You
Yes,
only have three.
yes.
So all is cis to trans to cis.
From trans,
to cis.
from trans,
Yes.
yes.
Cis only have one carbon, right?
From cis to trans, only one
Yeah.
happen. But from trans, there are three possible. There are three pathways. And in here, actually, from condom is just kind of center of the peak. And some trajectory wants to go C11 shift after the photoatomization because it's symmetric. It's kind of it's planar system. So there could be some thermal fluctuation which force the trajectory go to the C11 shift. However, there are several state interactions which hinder the path for that. So, and it cannot go here. So this path has to wander to find out the path, the proper path. And during this wandering process, this coherence is lost because some paths can go this way directly.
So basically, you say your initial correlate and reaction correlate is not in the C setting.
Right.
It's merely like
right.
searching along.
Right.
the
That's right.
Okay.
This is the mechanism for losing the coherence. In rhodopsin, however, there are strong, force or how to say, strong tendency to go to the proper path. That's why every path is going to this imaging.
Okay, quickly, another question is, when you do this rhodopsin simulation, did you picture match recently masses data?
Actually,
Raman data?
Yes. Actually, I have some. I expected that question, and Actually, yes and no. And we found very large excitations of food mode, which is called, which are which are consistent with matrix matrix experiment. And also we calculated the time dependent shift of this frequency mode. I don't have that figure. However, what he proposed based on his measure is that first poop motion is largely governed, and then the photoatomization is followed by following that food motion. However, we don't have such kind of large food excitation on the excited state. The food mode excitation is just after the photoatomization, just after the intersection, conical intersection. So we don't agree with their model, but we certainly agree with their data.
Right, I think we better draw the discussion to a close now and move on to our next talk. So let's Right, and the next speaker is Seth Olsen. This really is the Seth Olsen day, and I did suggest to him earlier, but perhaps you might like to give an after-dinner speech, just round it all off, humorous, quick about what you might just get married.
You'll be the best man of the day.
So Seth's going to share with us some of his work on the bridge isomerization of fluorescent protein 24 models. I'll stand up 5 minutes before the end and stand up here. So,